I'm Abhay Dandekar. I'm a professor at the University of California in Davis in the Plant Sciences Department. And our lab has been working on Pierce's disease, trying to understand what causes the disease symptoms. And it was generally believed that Xylella grows in the xylem and limits the flow of water. And we examined proteins that are secreted that may be responsible for the virulence response, and have been characterizing those proteins. And we stumbled upon one of these proteins that is a lipase that breaks down lipid molecules that appears to be responsible for causing the typical symptoms, scorching, edge scorching symptoms that one observes with Xylella. The breakthrough came when we began examining proteins that are secreted and characterizing them, and we came across this highly abundant, previously uncharacterized protein that was secreted by Xylella that we found associated closely with the symptoms of Xylella. And we then were able to establish a cause and effect relationship between the symptoms and the presence of this protein.
So this is a plate, and this is a bacterial plate, and this is how we detect the enzymatic activity of LES-A, the virulence factor that we identified. We put them in a emulsified agar plate, and as the lipase or LES-A enzyme hydrolyzes the fat in the plate, we see a clearance zone within the culture.
Xylella, our bacteria, actually has a lot of different proteins that it puts into the environment around it. And one of these proteins, LES-A, has a certain activity that it does that we can detect in a machine. And in that machine, our protein will break down a certain chemical, and then when it breaks it down, it'll light up in the machine, and that the amount of light that it produces we can measure, and that way we can see how much activity our protein is having.
Another way we can test the activity of the LES-A protein is to extract it from bacteria and then suspend it in buffer and inject it into plant tissue and observe the level of symptoms. And the way we do that is we use a syringe. This syringe now contains our protein, and we infiltrate it into a leaf and then we will be able to observe whether this particular protein creates any stress or necrotic lesions that we see associated with Pierce's disease.
I think it's really important because if we understand the virulence mechanism, or the mechanism by which Xylella causes disease, then we can blunt those processes, develop diagnostics that can identify infections at early stages, also try to develop other strategies to cure the disease.